Hello there again. Good day. So for today, I'll be discussing antibody structure and functions. We are on chapter 4 and I am still using Clinical Immunology and Serology by Stevens, and this is the third edition. Okay, so when B lymphocytes are stimulated by antigen and undergo differentiation, the end product is antibody, or we call it immunoglobulin. This is the picture of immunoglobulin, and I get this from the link below. Immunoglobulins are actually glycoprotein found in the serum portion of the blood. They are composed of 82% to 96% polypeptide and 2% to 14% carbohydrates. Now, when subjected to electrophoresis at a pH of 8.6, appears primarily in the gummy band. And your immunoglobulin is divided into 5 classes. We have the IgA, IgM, IgG, IgD, and IgE. Now, I have these mnemonics in order for me to remember it always, and I usually call it the GAMED. So, IgG, IgA, IgM, IgE, and IgD. Okay, so let's try to erase this first for a moment. Okay. Now, your immunoglobulins or your antibody are considered to be the humoral branch of the immune response. Now, they play an essential role in antigen recognition and in biological activities related to the immune response, such as we have opsonization and complement activation. Now, although each class has unique properties, all immunoglobulin molecules share many common features, and we'll be discussing each of them as we go along with our discussion. So for this slide, we have the structure of an antibody. All immunoglobulin molecules are made up of a basic 4-chain polypeptide unit that consists of 2 large chain, this one, we call it the heavy chain or the H chain, and 2 smaller chain we call the light chain or the L chain. And these chains are held together by non-covalent forces and disulfide bonds. This one, disulfide bridges. Okay, I will try to erase it first so we can see the illustration clearly. Okay, there we go. Okay, so the basic structure of immunoglobulin was elucidated in 1950s and 1960s by the efforts of 2 men. We have Gerald Edelman and Rodney Porter. Gerald Edelman, working at the Rockefeller Institute in the United States, and Rodney Porter at Oxford University in England, and for their contributions, these 2 men shared what we call the Nobel Prize, okay, in physiology and medicine, and that is 1972. Okay, so I will try to erase first so we can see everything clearly. Okay, for a moment. I guess this one is okay now. Here's another one. Okay, I guess that's good enough. Now, Edelman's work centered on using the analytical ultracentrifugation to separate out immunoglobulins on the basis of molecular weight, while Porter's work was based on the use of proteolytic enzyme, which is papain. Edelman found that the intact IgG molecules has a sedimentation coefficient of 7s. Now, the S here, this is the, we call it the Speedberg unit. Okay, indicates, and this one, the Bidford unit, indicates the sedimentation rate in an analytical centrifuge. Okay, so larger molecules will travel farther and thus have a larger sedimentation coefficient. Okay, that is according to Edelman. Now, on obtaining a purified preparation of IgG, Edelman this time used 7M urea. Use 7M urea, okay, to unfold the molecules. Now, once unfolded, let me erase this. I'm a little bit distracted by this one. Okay. Once unfolded, the exposed sulfhydryl bonds could be cleaved by a reducing agent called your mercaptoethanol. Mercaptoethanol. Okay, so this is the reducing agent. Again, the mercaptoethanol as a reducing agent. After such treatment, the material was subjected again to ultracentrifugation and two separate fractions. Okay, so in the next slide, we can see the two separate fractions. We have the H chain here. Oh, sorry, this is the H chain with 3.5 S structure. Again, the S there is the Bidford unit of coefficient with a molecular weight of 50,000, while the L chain here has 2.2 S fraction with a molecular weight of 22,000. Now, these two pieces occurred in equal equations or equal amount, and indicating the formula for your IgG, which is the H2L2, and actually this is the generalized formula for all immunoglobulins. So this slide represents the generalized structure of immunoglobulin. So as a recap, the basic structure of immunoglobulin is a tetrapeptide consisting of two H chain here, or we call it the heavy chain, and two light chain, or we call it as well as the L chain. And these two H chain and L chain is linked by a disulfide bonds, and so we have here the interchain disulfide bonds. Okay, but before we'll proceed, we'll try to erase first so we can see the structure clearly. Okay, there we go. Now continuing, the amino terminal end of each chain here is a variable region, while this carboxy terminal end is the constant region. We also have a pepsin digestion, which yields a Fab, Fab2, or we call it the fragment antigen binding here. Okay, with all the antibody activity, of course, and the FC1, or we call it the fragment crystallizable region. Okay, and while the papain cleavage site or papain digestion yields two Fab fragments and an FC portion. Aside from that, somewhere in this area, you can see the hinge region, which is located in between the CH1 and the CH2 region, which we call it the hinge region. The hinge region is or has a high content of proline and hydrophobic residues. Now, the high proline content allows for flexibility. Okay, well, we also have here, we'll erase again for a moment. Okay, there we go, and maybe it would be better to erase so will not be distracted by all of those writings. Now, we also have the intrachain disulfide bond here, which creates a flooded region, or we call it the domain. Okay, then we have the variable region and the constant region. So this is the generalized structure of your immunoglobulin. So again, very quickly, this slide is the annotated diagram of immunoglobulin structure. As you can see, this is the IgM, IgG, and IgA structure. In this area here, this is the antigen binding site. And then this is the hinge region, which allows for flexibility, and that is due to high proline content. Okay, and of course we have the legend. This one is the hypervariable region. This is your heavy chain, and this is the light chain. This is the variable region here, and we have the constant region here. Both the heavy chain and the light chain is being linked by a disulfide bond. Okay, and then somewhere here, these are the interchain region, and then of course the intrachain region creates a flooded region, or the domain. And of course this is where your CH2 and CH3 occur. So this is the constant region. Okay, now let's talk about cleavage with papain. Well, I was saying a while ago, Porter's work was based on the use of proteolytic enzyme papain, which was used to cleave IgG into three pieces of about equal size, and each having a sedimentation coefficient of 3.5 S and representing a molecular weight of approximately 45,000 to 50,000 Daltons. Now, the carboxyl cellulose ion exchange chromatography separated this material into two types of fragments, one of which spontaneously crystallized at 4 degrees centigrade. Thus, this fragment is known as your Fc fragment, or we call it the fragment crystallizable. Now, this Fc fragment is important in effector functions of the immunoglobulin molecules, which includes opsonization and complement fixation. Now, this fragment, the Fc fragment, had no antigen binding ability and is now known to represent the carboxyl terminal halves of two heavy chains that are held together by the disulfide bonding, while the remaining two identical fragments were found to have antigen binding capacity and were named as your Fab fragments, or again the fragment antigen binding. Now, because precipitation would not occur if Fab fragments were allowed to react with antigen, it was guessed that each fragment represented one antigen binding site, and that two such fragments were present in an intact antibody molecule, such as a molecule would be able to form a cross-linked complex with antigen, and the complex would precipitate. So each fragment is consist of the 1L chain and 1/2 of an H chain held together by disulfide bonding. So this is the Fab fragment. This is the disulfide binding. Okay, so again, each Fab fragment is consist of 1L chain, like this one, and 1/2 of the heavy chain held together by the disulfide bonding. So this is the work of the cleavage with papain. Now, after papain digestion, let's talk about pepsin digestion. So Alfred Misanoff used pepsin to obtain additional evidence for the structure of immunoglobulin. Now, this proteolytic enzyme was found to cleave IgG at the carboxy terminal here. It leaves carboxy terminal side of the interchain disulfide bond, thus yielding this one. It cleaves the carboxy terminal side, yielding one single fragment with a molecular weight of 100,000 Dalton and all antigen binding ability known as the, what we call that again, the Fab2 region or antigen binding fragments. An additional fragment called the FC, which is a little bit similar to the FC or the fragment crystallization, except that it disintegrated into several smaller pieces. Thus, a basic picture of the 4-chain unit of the immunoglobulin molecule was obtained, which indicated that each light chain or L chain was bonded to a heavy chain or H chain by means of the SS band. Now, as you can see, this one, this is the L chain, and then this is the H chain or the heavy chain, then it is bonded by a disulfide bond, and of course the heavy chain were joined to each other by one or more disulfide bond. Again, now if you can see this one, so the exact number of disulfide bonds differs among antibody classes and subclasses. So for this particular slide, we'll just have a quick review on the basic antibody structure, particularly explaining the papain and pepsin digestion. So let's start with this illustration. Now, as you can see, the papain cleavage here, this is based on Porter's work, which he uses proteolytic enzyme papain, which was used to cleave IgG into three equal sizes and each having a sedimentation coefficient of 3.5. That's a Speedberg unit representing a molecular weight of approximately 45,000 to 50,000 Daltons. Okay, so let's try to erase first, so we can still write on here somewhere later. Okay, now carboxymethylcellulose ion exchange chromatography separate this material into two fragments. We have the FC fragment and the Fab fragment. Okay, so your papain cleavage will eventually cut on this interchain bond here, while your pepsin cleavage, as you can see, which is the work of Alfred Nisonov. He uses pepsin to obtain additional evidence for the structure of immunoglobulin. Now, the pepsin, which is a proteolytic enzyme, was found to cleave at the carboxy terminal here, at the carboxy terminal side of the interchain disulfide bond. So, this is for the cleavage area for the pepsin, and this is the cleavage area for the papain. Okay, then let's discuss the nature of the light chain. Now, the difficulty in obtaining a significant amount of specific immunoglobulin for amino acid analysis was overcome by the discovery of what we call the Bence-Jones protein. Okay, this Bence-Jones protein is found in the urine of a patient with multiple myeloma, where in fact the L chain that were being secreted by the malignant plasma cells. Bence-Jones protein had been discovered in 1845 by Dr. Henry Bence-Jones, thus the name Bence-Jones protein, who noted the peculiar behavior of this protein. Now, when heated to 60 degrees, so if you're going to heat it to 60 degrees, they precipitate from urine, but on further heating to 80 degrees, they redissolved, and this characteristic made it possible to isolate the L chain and obtain the amino acid sequence. Okay, so let's erase again. prior to continuing with our discussion. So we can still write if there's need to write. Okay, thank you so much. Then the analysis of several Benz-Jones protein revealed that there were actually two main types of the L chain, which is known as the kappa and the lambda chain. So now each contains between 200 and 220 amino acids, and from position number 111 on the amino terminus, which is position number 1. Now, it was discovered that each type had essentially the same sequence. Now, this region was called the constant region, and the amino terminal end was called the variable region. So this is called your variable region, and this one is the constant region. Thus, all kappa light chain have an almost identical carboxyl terminal end, and the same is true of the lambda chain. Now, the difference between the kappa and lambda chains lies in amino acid substitutions at a few locations along the chain. So there are no functional differences between two chains. So both kappa and lambda chains are found in all 5 classes of immunoglobulin, but only one type is present in a given molecule. The heavy chain sequencing demonstrated the presence of domains similar to those in the light chain. So if this is your light chain, then this is the heavy chain here. Okay, so that is it has also the variable region and the constant region here. So allow me to erase first so that we can clearly see the illustration here. Okay, that's it. Okay, now the first approximately 110 amino acids at the amino terminal end constitutes the variable region here, and the remaining amino acids can typically be divided into 3 or more constant regions with very similar sequences designated as your CH1 or constant heavy chain 1, like here, and the CH2 or the constant heavy chain 2, and the CH3 or constant heavy chain 3. Now, the constant region of the H chain are unique to each class and give each immunoglobulin type its name. Hence, we have IgG, okay, has a gamma chain, IgM has a mu chain, your IgA has alpha chain, your IgE has epsilon chain, and the IgD contains the delta chain. Okay, so each of these represents an isotype, a unique amino acid sequence that is common to all immunoglobulin molecules of a given class in a given species. Minor variations of these sequences that are present in some individuals but not others are also known as your allotypes, and the allotypes occur in the 4 IgG subclasses, in 1 IgA subclass, and in the kappa light chain. Now, these genetic markers are found in the constant region and are inherited in simple Mendelian fashion. So some of the best known example of allotypes are variations of the gamma chain known as we have G1, M3, and the G1, M17. Okay, so however, allow me to erase this one again for a moment. This one, okay, that one. Now, the variable portion of each chain are unique to a specific antibody molecule, and they constitute what is known as the idiotype of the molecule. Now, the amino terminal end of both the light chain here and the heavy chain contains these regions, which are essential to the formation of your antibody binding site. Now together they serve as antigen recognition unit. So in this illustration, here you can see the hinge region, this one. So this is a segment of the heavy chain located between the constant heavy chain 1 and constant heavy chain 2. So this is your hinge region. Now, it has a high content of proline and hydrophobic residues, and the high proline content allows for flexibility. Now, this ability to bend lets the two antigen binding site, or the Fab portion of the antibody, to operate independently, and flexibility also assists in effectors functions such as initiation of the complement cascades. Now the gamma, the delta, and the alpha chain all have a hinge region, but your M, or the mu chain, and the epsilon chain, okay, does not have uh, the hinge region. However, the CH2, or constant heavy chain 2 domains of these mu and epsilon chains are paired in such a way to confer flexibility for the Fab arms. Now, in addition to the 4 polypeptide chains, all types of immunoglobulin contains a carbohydrate portion which is localized between your constant heavy chain 2 domains of the two heavy chains. Now, functions of the carbohydrates includes number 1, increasing the solubility of immunoglobulin. So again, increase solubility of immunoglobulin. This is one of the function. Second is it provides protection against degradation. The third, it's enhancing functional activity of the Fc domain, and basically the third function may be the most important because recognition by Fc receptors correlates with the presence of the carbohydrates moiety. Okay, so let's discuss the specific immunoglobulin classes, and let's start with IgG, or immunoglobulin class G. Now IgG is the predominant immunoglobulin in humans, comprising about 75 to 80% of the total serum immunoglobulins. It has the longest half-life of about 23 to 25 days, which of course may help to account for its predominance in the serum. Now your IgG is divided into 4 major subclasses with the following distribution. So for IgG1, it accounts for 67%, and your IgG2 accounts for 22%, while the IgG3 accounts for only 7%, and IgG4 accounts for 4%. Now, these subclasses differ mainly in the number and position of the disulfide bridges between the gamma chain. Variability in the hinge region affects also the ability to reach for antigen and complement activation. Now, let's try to erase first. Okay, so for IgG3, it has the largest hinge region. So as you can see, this is the hinge region, and the largest number of interchain disulfide bonds. So this is the hinge region, and this is the disulfide bond here, this one. Okay, so again, this is the hinge region. Okay, now since it has the largest number of interchain disulfide bond, therefore it is the most efficient at binding complement, and this is followed by your IgG1, then we have IgG2, and lastly IgG4. Now let's proceed Okay, so for the major function of IgG, we have number one, providing immunity for the newborn because IgG can cross the placenta. Although all subclasses of IgG appears to be able to cross the placenta, IgG2 is the least efficient. Second, it also helps in fixing complement. And third, coating antigen for enhanced phagocytosis or opsonization. So we have macrophages, monocytes, and neutrophils have receptors on their surfaces that are specific for the Fc region of the IgG. Now, this enhances contact between antigen and phagocytic cells and generally increases the efficiency of phagocytosis. Now, although your IgG1 and IgG3 are particularly good at initiating phagocytosis because they bind most strongly for Fc receptors. Well, the fourth one is neutralizing toxins and viruses. So IgG has a high diffusion coefficient that allows it to enter extravascular spaces more readily than other immunoglobulin types. In fact, it is distributed almost equally between the intravascular and extravascular spaces. Thus, the IgG plays a major role in neutralizing toxins and viruses. And lastly, IgG participates in agglutination and precipitation reaction as well. So agglutination and precipitation reaction takes place in vitro, although it is not known how significant a role this plays in vivo. Well, the IgG is better at precipitation reaction than at agglutination because precipitation involves small soluble particles which are more easily brought together by the relatively small IgG molecules. Agglutination is, on the other hand, is the clumping together of larger particles such as red blood cells, and being a larger molecules, we have say IgM is much more efficient at at this than the IgG. So these are the functions of the IgG. So next of the immunoglobulin class is the IgM, which is known as macroglobulin because it has a sedimentation rate of 19S, or the Svedberg unit, which represents a molecular weight of approximately 970,000. Now the half-life of IgM is about 10 days, which is much more shorter than the IgG. It accounts for between 5 and 10% of all serum immunoglobulins. If IgG is treated with mercaptoethanol, it dissociates into 5 7s units, each having a molecular weight of 190,000 and a 4-chain structure that resembles IgG. For the molecular weight of the heavy or the mu chain is approximately 70,000, and it is consists of 576 amino acids and includes one or more constant domain that is found on the gamma chain. Now the pentamer form, so this is the pentamer form is found in secretions, while the monomer form occurs in the surface of the B cell. Now, the 5 monomeric units are held together by what we call the J or joining chain, which is a glycoprotein with several cysteine residues. This serves as linkage point for disulfide bond, your J chain, between two adjacent monomers. Linkage actually occurs at the carboxy terminal end of two of the mu chain, and it appears that the J chain may initiate polymerization by stabilizing the Fc sulfhydryl groups so that cross-linking can occur. Now, the molecular weight of the J chain is approximately here 15,000, and one J chain is present per pentamere. Now, the IgM thus configures assumes a star-like shape, just like this one. This is a star-like chain, shape with 10 functional. It has 10 functional binding site. Okay, 10 functional binding site. Only about 5 of these are used unless the antigen is extremely small. Okay, so let's try to erase that first. Okay, now let's continue. The high valency of IgM antibodies contravenes the fact that they tend to have a low affinity for antigen. Because of its large size, IgM is found mainly in the intravascular pool and not in the other body fluids or tissues. Because it cannot cross the placenta, IgM is known as the primary response antibody. Again, it is known as the primary response antibodies. Okay, and because it is the first to appear after antigenic stimulation, and it is the first to appear in the maturing infants. Well, it is synthesized only as long as antigen remains present, because there are no memory cell for IgM. Now, depicts the difference between the primary response, which is predominantly IgM, and the secondary response, which is mainly IgG. Now, the primary response is characterized by long lag phase, while the secondary response has a shortened lag period and much more rapid increase in antibody titer. Okay, now as with regards to the function of the IgM, number one, of course, it's for complement fixation, which is the most efficient in triggering the classical complement pathway. Okay, and second, agglutination. The larger number of binding site makes IgM more efficient in agglutination reaction. And then we also have the opsonization, in which IgM forms a potent defense against many bacterial diseases. And lastly, toxin neutralization. So those are the function of IgM. Well, IgM forms a potent defense against many bacterial disease because IgM has a J chain, and it can occasionally acquire a secondary complement like IgA does, and this allows to be traverse epithelial cells and patrol mucosal or mucous membrane. IgM also serves as a surface receptor for antigen. Now, in the cytoplasm of the pre-beta cell, mu chain first appear. When they associate with the early surrogate L chain, a sign is sent to exclude rearrangement of the other heavy chain glucose and to begin rearrangement of the gene controlling light chain synthesis. Now later, as L chain or light chain are synthesized, IgM monomers are formed and become inserted into the plasma membrane, and the process of membrane IgM classes the lymphocytes as mature B cells. Okay, so that is the function of the IgM. Now let's proceed with the IgA. So in the serum, IgA represents 10 to 15% of all circulating immunoglobulin, and it appears as a monomer with a molecular weight of approximately 160,000. It has a sedimentation coefficient of 7s, and migrates between the mu and the beta region on electrophoresis. Now, the heavy chain, called the alpha chain, has a molecular weight of about 55,000 and 60,000, and it is consists of 472 amino acids. There are two subclasses designated as IgA1 and IgA2. Now these two subclasses differ in content by 22 amino acids, 13 of which are located in the hinge region and are deleted in IgA2. Now the lack of these region appears to make IgA2 more resistant to some bacterial proteinases that are able to cleave IgA1. Hence, IgA2 is the predominant form in secretion at mucosal surfaces, while IgA1 is mainly found in the serum. Furthermore, IgA is found as a dimer along the respiratory, urogenital, and intestinal mucosa, and it appears in milk, saliva, tears, and sweat. Now, since mucosal surfaces are a major point of entry for pathogens, IgA2 serves to keep antigen from penetrating further into the body. Now, the dimer consists of two monomers held together by a J chain. Here, this is the J chain that has a molecular weight of about 15,000. Secretory IgA, on the other hand, is synthesized in plasma cell, found mainly in mucosal associated lymphoid tissue, and it is released in the dimeric form. Now, IgA is synthesized at a much larger or much greater rate than that of IgG, like approximately 3 grams per day in the average adult, but because it is mainly in secretory form, the serum concentration is much lower. Now, a secretory component SC, which has a molecular weight of about 70,000, is later attached to the Fc region of the antibody around the hinge portion of the alpha chain. Now, this protein, consisting of 5 immunoglobulin-like domains, is derived from epithelial cells found in close proximity to the plasma cells. The secretory component, with a molecular weight of 100,000, is actually found in the surface of the epithelial cells and serve as a specific receptor for IgA. Now, the plasma cells that secrete IgA actually home to subepithelial tissues, where IgA can bind as soon as it is released from the plasma cells. Now, this homing of activated lymphocytes depend upon a high level of certain adhesion molecules that allow binding to epithelial cells. Once binding takes place, IgA and secretory component precursor are taken inside the cell and then released to the opposite surface by a processing known as transcystosis. So we have transcystrosis. Now, the vesicle carrying IgA and the secretory component receptor fuses with the membrane on the cell's opposite side, and a small fragment of SC or secretory component is cleaved to liberate the IgA dimer with the remaining secretory component. Thus, the secretory component may act to facilitate transport of IgA to mucosal surfaces. It also makes the dimer more resistant to enzymatic region by making sites that would be susceptible to protease cleavage. Now, for the main function of the secretory IgA, First is to patrol mucosal surfaces and act as a first line of defense. Now, it plays an important role in neutralizing toxins as well produced by microorganisms, and it helps to prevent bacterial adherence to mucosal surfaces. So complexes of IgA and antigen are easily trapped in mucose and then eliminated by the ciliated epithelial cells of the respiratory or intestinal tract. This prevents pathogens from colonizing the mucosal epithelium. Since IgA is found in breast milk, breastfeeding helps to maintain the health of the newborns. Okay, and it appears that IgA is not capable of fixing complement by the classical pathway, although aggregation of immune complexes may trigger the alternate complement pathway. Okay, now lack of complement activation may actually assist in clearing antigen without triggering an inflammatory response, Now, thus minimizing tissue damage. Additionally, the neutrophils, monocytes, and macrophages possess specific receptors for IgA. Binding to this site triggers a respiratory burst and degranulation, and this occurs for both serum and secretory IgA, indicating that they are capable of acting as an opsonins, and the success of oral immunizations such as the Sabin vaccine, which induces IgA almost inclusively, demonstrates the effectiveness of IgA pro, um, protective role on mucosal surfaces. Well, so for IgG, on the other hand, it was not discovered until 1965. So 1965, when it was found in the patient with multiple myeloma. IgD is extremely scarce in the serum, representing less than 0.001% of the total immunoglobulins. It is synthesized at a low level and has a half-life of only 2 to 3 days. Now, the molecules has a molecular weight of approximately 180,000, and it migrates at a fast gamma protein. Now, the delta heavy chain has a molecular weight of 62,000 and appears to have an extended hinge region consisting of 58 amino acids. Now, most of the IgD present is found in the surface of immunocompetent but unstimulated B cells or B lymphocytes, It is the second type of immunoglobulin to appear, IgM being the first, and it may play a role in B cell activation. Now, the high level of surface expression and its intrinsic flexibility make it an ideal early responder to antigen. Thus, cells bearing only IgM receptor appears incapable of an IgG response. Now, while those with both IgM and IgD receptors are capable of responding to T cell help and switching to synthesis of IgG, IgA, and IgE. Thus, IgD may play a role in regulating B cell malnutrition and differentiation. Now, for IgG, because of its unusual long hinge region, IgD is more susceptible to proteolysis than other immunoglobulin. Now, it does not appear to serve a protective function because it does not bind complement, and it does not bind to neutrophils or macrophages as well. Aside from that, IgG does not cross the placenta. Now, because of the unusual long hinge region, IgD is more susceptible to proteolysis than other immunoglobulins. Um, this is maybe the main reason for its short half-life, and in the secreted form in the serum, IgD, does not appear to serve a protective function because it does not bind complement and it does not bind to neutrophil macrophages, and it does not cross the placenta as well. And the last one is the IgE. So it is the least abundant immunoglobulin in the serum, accounting for only 0.0005% of total serum immunoglobulin. IgE is an 8S or Svedberg unit molecule with a molecular weight of approximately 190,000. Now the kappa or the heavy chain is composed of around 550 amino acids that are distributed over one variable and four constant domains. Now, a single disulfide bond joins each kappa chain to the light chain, and two disulfide bond link the heavy chains to one another. IgE is most heat labile of all immunoglobulins. Heating to 56 degrees centigrade for between 30 minutes and 3 hours result in the conformational changes and loss of ability to bind to target cells. Furthermore, IgE does not participate in typical immunoglobulin reactions such as complement fixation, agglutination, or opsonization. Additionally, it is incapable of crossing the placenta. Instead, shortly after synthesis, it attaches to basophil and tissue mast cells by means of specific surface protein, which is termed as high affinity Fc epsilon R1 receptor, which are found exclusively on these cells. The molecule binds at the constant heavy chain 3 domain on the Fc region, and this leaves the antigen binding site free to interact with specific antigens. Plasma cells that produce IgE are located primarily in the lungs and in the So the function of IgE. Plasma cells that produce IgE are located primarily in the lungs and in the skin. Now, mast cells are also found mainly in the skin and in the lining of the respiratory and alimentary tract. One such cell may have several 100,000 receptors, and each is capable of binding IgE molecules. Now, when two adjacent IgE molecules on a mast cell bind specific antigen, a cascade of cellular events is initiated that result in degranulation of the mast cell with the release of vasoactive amines such as the heparin, and the other one is the histamine. Okay, now release of these mediators induces what is known as, we call it type 1 hypersensitivity. Hypersensitivity. Okay, or we call it as well as allergic reaction. Now, typical reactions includes hay fever, asthma, vomiting, and diarrhea. So have hives and also the life-threatening anaphylactic shock. Now, while IgE appears to be a nuisance antibody, it may serve a protective role by triggering an acute inflammatory reaction that recruits neutrophils and eosinophils to the area to help destroy invading antigens that have penetrated IgA defenses. Eosinophil especially plays a major role in the destruction of large antigens such as parasitic worms that cannot be easily phagocytosed, like what we've seen in this illustration. Now we have a helmet And since it is a very large um, antigen or a foreign substance for that matter, well, eventually the IgE will recruit eosinophils and some other neutrophils to the area, then it will eventually help destroy the antigens that have penetrated, okay, IgA defenses. So this is the action of the function, I mean, of the IgE. Thank you for watching, and that's the end of our lecture. I hope that you learned something from this topic. Have a good day.